Good evening, everybody. I think we're about to start the Science A core night before the exam hangout. And again, hopefully you should be able to hear me at this point. Now, this is a slightly different hangout to the previous ones. We're not actually going to be going through any of the content. It's simply a chance to hopefully put you at ease for the exam tomorrow, so you know exactly where you stand. And my objective is to hopefully go through some of the key things that you're going to find in the exam and how you're going to tackle those particular questions tomorrow. Again, if you're on via Frog, please just post any questions as I go through them, and I'll do my very best to answer. So let's begin. Well, the key topics for the core science are listed below. And as I said at the start, I'm not going to be spending time going over the content. Roughly speaking though, you should be aware of diet, disease, nerves and hormones, homeostasis and drugs, DNA, the environment, and finally, life on Earth. Now, a precursor to all of this, we're clearly unsure of what exactly is going to be asked in tomorrow's paper. But we can have a rough idea of things that are likely to come up. With that in mind, obviously, we don't know specifically the questions that will be coming up. So the safe answer is you should be aware of all of these parts of the content. However, over the years, I would say the following. Disease comes up every single year. If that's bacteria, viruses, vaccinations, potentially growing organisms. Nerves and hormones is a funny one. It's normally one or the other. Again, it's difficult to put my money on. I'll be inclined to make sure that I know both quite clearly. There are normally a few questions on homeostasis and drugs. It's normally around the testing of drugs or the use of, say, statins or steroids. DNA is, again, very common. Last year was cloning. Perhaps this year it would be genetic modification. Again, maybe safe to look at both. Adaptations are normally there every single year. How an animal is adapted to where it lives. Decay, carbon, normally one of these two. And Darwin, with the theory of natural selection and evolution, is something that comes up every year. I've left out diet. I think diet this year might be a big mark. It's not often in the papers for more than a few marks, but there's a lot in the news recently about the sugar tax linked to diabetes, which kind of links us into our homeostasis about insulin production, potentially even DNA in genetically modifying insulin. So I think for once, diet might also play a key part. But we won't go into the specifics. Hopefully your knowledge at this point is pretty good. The plan for me tonight is essentially just to get you prepared and ready for the exam. Well, here's a few basic fundamentals, which are absolute common knowledge and sense, but can easily be overlooked. The first thing is eat some food. Have a really good breakfast, take a lunch. Don't rely on queuing up at the canteen in case you're going to be late for it. Have a big bar of chocolate, whatever it is that you need to get through the exam, just make sure you've got lots of food in your belly. And also, if it's going to be a relatively hot day again tomorrow, make sure you're hydrated. Plenty of water, Perhaps you've got a water bottle that you've taken to school. All the basics, just be prepared. Second of all, sleep tonight. Tonight is not one of those evenings where you're up until 3 p.m. You're certainly not up during the night worrying. It's going to do you no good. I'm sure you've done the work for this. I'm sure you're prepared. So try and get a really good night's sleep. Thirdly, obviously, but you wouldn't be the first if you didn't, turn up. Arrive to school promptly. It's the exam either at 9 a.m. or 2 p.m. Make sure you're there for them. Make sure you kind of know where you're going, be that the hall or in a particular area. Finally, one thing to check tonight, have you got your equipment? You need a couple of pens in case one breaks, a pencil, a ruler, a rubber, calculator. It's a biology exam tomorrow, but take it. There are going to be some maths questions I'm going to show you later on. Take a calculator, make it easy for yourself. So that's preparing yourself for tomorrow. When you're there, these are all basic, but know where you're going to be sitting. Know the room that you're in and the start time. Look at the marks. As soon as you get your paper, look how many you've got. Now, I can already tell you in advance what it's going to be out of. It will be out of 60. It's an hour's paper. Have that in your mind. By the time you are 30 minutes through, you should be about halfway through the paper. Ideally, a little bit ahead of that. So just be aware you know where you are. Give yourself time. Look at the clock in the exam room. Know where you are. Know when you started. Know what time you finish. Know where you should be. And ideally, you want about 5 minutes at the end to go over your questions. Check the marks. Each question will have a certain number of marks associated to it. And the rule of thumb, every single mark needs a different point. Don't include silly comments. The examiner will not find them funny. They are unlikely to do you any favours if you're not helping yourself. But also, don't leave blanks. Make an educated guess at worst. It's better than leaving a blank. Absolutely don't be writing, I don't know. That's going to get you 0 marks. It's going to be a waste of your time, and it's probably going to annoy the examiner. So do your best, an educated guess on all questions. Now, this, of all the important points, is probably the most. You've got yourself right now, if you've treated these exams seriously, a load of content in your head. And one of the worries is, are you going to forget it on the day? Are you going to get to question 2 and suddenly have a mind blank? Well, Dr. Fellis and I have been in discussion with this, and we really think it's probably quite prudent to do a very quick brain dump at the start of each question. What we mean by this is as follows. Question 1 will be on something. Let's call it DNA. And it will have part B, part C, part D, etc. Question 2 might be on Darwin. And it will have A, B, C. And question 3 could be on something such as adaptations. When you get to the start of each question, so perhaps you turn the book and you see the very first question, question 1, suggests to you that it's on DNA, write down everything you know. Genes, variation, sexual, asexual, anything you know that's going to trigger your mind for the subsequent questions. And the same with Darwin. He went up against Lamarck with conflicting theories. It was natural selection. There were arguments around it with God. Adaptations, prey, predator, camouflage. So take that instant brain cleanse. Write down what you know on that very first question around the side of it. So in the top space, in some white space, anywhere that you can find, even on the back. But the key thing, you only really want to be doing that for about 30 seconds. There's roughly going to be about 8 questions. So if you did a 30-second brain dump for the very big question, not the part B's or part C's or part D's, but the actual first part of the question that gives you a clue as to what it's going to be asking, if you do that, there's about 4 minutes already gone. Now, we think it's really valuable, but don't spend too long. Don't spend 5 minutes writing down everything you know on the subject. You will run out of time. Final part, look at the question, because it often refers back to part A, part B, and it happened last year, there was a question, part D, where the answer to it was written in the text in part A. So it gives you some clues. Now, the final bit out of all of this, watch how you write. Slightly ironic, because I'm trying to write here, and as a few people have commented, my writing, as well as my drawing of dinosaurs, is pretty poor. Do your best to make your writing as clear for the examiner. With the exception of the spelling, punctuation, grammar question, if you've got pretty messy writing, if you've got lovely writing that is really, really difficult to comprehend, go for some capitals. Take your time. Hopefully, your writing will be clearer than mine on this electronic board. So there are our top tips. Now, the remainder of this particular session will be looking through some example questions. And I'm going to show you, hopefully, where the clues are and how to answer the specific ones. Just before I move on, a quick question that's come up. Are you likely to get marked for spelling, punctuation and grammar on all 6-mark questions? They will be very clear. I'll quickly run to the question and then we will come back to it. You'll always be given this kind of information. If they are looking at your spelling, punctuation, grammar, they will tell you they are. Now, your spelling needs to be ideally accurate in any other non-SPG question, but if the examiner knows what you are essentially suggesting, you'll probably get the marks. So, for example, if the question was something like, who came up with the theory of natural selection, and rather than spelling Darwin, you spelled him Darwin, that might be a little bit too far out, but something like that, you're probably getting the mark. Okay, let's go back. So let's have a look at the different contexts the questions come in, and then hopefully I'll talk you through where our points are coming from. So we're going to have a very quick look today at graphs, tables, the 6-marker, and then tick boxes or column boxes or circling answers. They tend to be more common in the foundation papers, but it's good practice to know where we're getting it from. And finally, diagrams. Again, this is not content heavy. If you're worried about content, just send us a comment. We'll do our best to answer it probably at the end. But today, for this session, for the next half an hour or so, where are we going to get our marks from? Okay, let's take a look. The very first set of questions I'm going to look at are the graphing questions. And hopefully, if your revision has been going well, these might look familiar because these have been taken from the 2015 papers. If you were sitting this exam last year, these are the questions you would have got. They range in higher and foundation papers, but the principle is the same. So we're well prepared. We go into our exam. We have our highlighter. We will do our best to supply a highlighter for you, but play safe. Find one, take one in. Figure 1, it's the impact on health caused by drugs from different sources. And then I've got a key. I've got my legally prescribed, my black one's here. I've got my legally non-prescribed, this slightly lighter grey, and then I've got my illegal, the lightest of all. Legal and non-prescribed drugs have a greater impact on health of the population than illegal. In other words, the legal, non-prescribed, we're thinking our minds alcohol, tobacco, they have a greater impact than our illegal ones, cannabis, heroin. Why? Well, let's have a look. Our first answer, and this is why I'm showing you today, is in the graph. This is the biggest part of the pie. So we have got the following. We've got the bigger impact. We're actually going to get a mark for stating what this pie chart shows us. The bigger impact. The other mark, and again, this is a kind of common sense mark, really, more people use them. Now that isn't in the graph, but that's just common sense. More people are drinking alcohol and smoking cigarettes than are taking heroin or cocaine. So we've got one mark from literally saying that is bigger than the other two. Our second mark for graphing becomes a little bit more complicated. We've got the mass of carbon in billions of tons per year. And we've got 2 points here. We've got one that is added to the atmosphere. In other words, carbon dioxide has been pumped out. And we've got one that is removed. And we go up to 150 either way. A tough graph to read this. Calculate the mass of carbon added to the atmosphere by respiration. Respiration is releasing carbon dioxide. It's going to be given out to the atmosphere. Now, we've actually got it here. We can have a look at the respiration of plants and algae, the respiration of animals, and the respiration by microorganisms. I would start this particular question by looking at my scale. I'm going from 0 to 50, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 small lines to get to 50. In other words, every small line is 5 billion tons. Well, then I simply need to add it up. And it's a pretty nasty question to do that because it isn't clear unless you did that first step. But clearly, respiration by algae is here. That's 50 tons. I've got myself respiration by animals, which is 50 and then 2 5s. So we're at 60, which is 110 so far. And then we've got our respiration by microorganisms, 5, 10, 15, 20, 25, 30. So I've got 140 billion tons. Nasty question. Start with the scale. Second point, some scientists are concerned that the mass of carbon in the atmosphere is changing. How does data in figure 2 support this idea? Well, again, we're tallying up. I've got 140 tons being released at this point by respiration, but I've actually got combustion here, which makes it a total of 150. 150 tons of carbon dioxide is going out to the atmosphere. This diagram here, I'm 2 small squares, which we know were 5 each, shy of 150. So actually, process X, X being photosynthesis, is 140. In other words, I'm taking in 140, but I'm giving out 150. I have a net of 10 billion tons of CO2 given out. That comes all from within this graph. I've used very little science. Yep, I knew process X was photosynthesis, and I knew the meaning of respiration and combustion, but the rest came from the graph. Okay, third example of a graph is this one. Now, I've shortened this than it was in the paper, but basically it comes in 2 forms. They give me a small table, which we'll look at later on in more depth, but then they give us a graph as well. So this is all about particular birds seen in the garden, the mean number. And then we've got the percentage of gardens in which the bird was seen. So I've seen on average 4 birds in 64% of the gardens. What I haven't told you, and I've missed it out from this exam question, but this data came from 2011, whilst this data here comes from 2012. The question is, calculate the percentage of gardens in which goldfinches were seen in 2012. Now, this is really tough. You might look at this and think, well, percentage of gardens is 80%. But subtly, it's not, because this is the change in the percentage of gardens in which the bird was seen. In other words, goldfinches have been seen 80% more than when they were seen in 2011. So with that in mind, if we've got ourselves 80% more, and in 2011 the percentage was 33.5, then 33.5 divided by 100 multiplied by 80, and that gives us, with my calculator, 26.8. If I add that then to my original 33.5, I get a total percentage of 60.3. A tough question, Matt, but look how I now check myself. If I'm saying in 2012 I'm 80% more goldfinches than I were in 2011, and in 2011 I had 33.5, really roughly in my head, if I double that number so it becomes 100% more, that answer would be 67%. So I know I'm looking for a number around about the 60% mark, certainly less than 67. So a tough question, this. That's where my calculator comes in useful, and that's why I'm really reading what the graph is telling me. Now, I've just had a very quick question that I didn't answer when we came to the spelling punctuation question. Where can we brain dump? Well, the honest answer is you can brain dump anywhere. What you've got to be careful is that you don't brain dump within questions. So if you've got a little bit of space, question 1, write your answer here. We don't want to be writing, obviously, in this bit, because that's where your answer's going. And you will see on the side of a paper a little column that says do not write. So try not to do it in there. But any other white space is yours, ignoring the front cover. So if you want to write on the back, you're going to see pages that say, with a big line in them, do not write on this page. What they're actually saying to you is, don't write your answers here. If you want to take a brain dump there, you go ahead. White space is yours. Just bear in mind, try not to write in any answer spaces, in a very clear side column where it says don't, and on the front bit where you need your official information. But if it's on the back, if it's on the side somewhere, you go ahead. That being said, when you take a brain dump, go for bullet points. You're not writing out lovely long essays, just get straight to the point for you to come back to. Okay, here's our last question on graphing. Again, this has all come from last year's paper. 3 to 4 graphs, 3 to 4 tables, a couple of diagrams, huge numbers of non-factual content, things that you need to analyse. Lots of marks available. Describe the effect of increasing the mass of cholesterol in the diet on the mass of cholesterol produced by the liver. To gain full marks, you should include data from figure 6, and this was figure 6. So let's take a look. The first thing that I'm drawn to is this key. I've got a straight line, which is blood cholesterol concentration, and I've got a hashed line, which is production in the liver. I've then got the mass of cholesterol in diet in milligrams per day. In other words, what's in my diet. And then I've got 2 different axes. I've got my concentration of cholesterol in blood, and I've got my mass of cholesterol produced by the liver. So there's lots going on in this graph. But naturally, I'm drawn to this. As I increase my mass of cholesterol in the diet, 2 things are happening. This one is going up. This one was my blood cholesterol. And there's no coincidence there. Funny enough, the more mass of cholesterol I have in my diet, the more I find in my blood. And the second one, along with massive cholesterol in my diet, I find that this particular one goes down. And this was my production, if I don't cross it out, production by the liver. Well, let's answer this. Very simply, as I increase my mass of cholesterol in my diet, I'm going to put the word C in bracket for cholesterol. As I increase my diet cholesterol, I find that my blood cholesterol also goes up. Quite slowly, but it does. And I also find that the cholesterol production by my liver goes down. Again, slowly. That's got me 1 mark. Hopefully you can see where the second mark's coming from, because something clearly happens here at 900. As I increase, as I go past 900 milligrams per day of cholesterol in my diet, I suddenly get a massive spike in blood cholesterol. It's no longer slow, it's fast, or it's high. And my liver production is no longer slowly falling, it falls quickly. There's a high rate of fall. You could at this point, and certainly if it was worth any more than 2 marks, if it had been 3 marks or so, you would have been including some numbers. So you would have been saying firstly, obviously, at the 900 point, but then perhaps the concentration of blood, well, the peak concentrations, what we're going to here. But it was only worth 2 marks. Again, a couple of points for 2 marks. Okay, that concludes the graphing point. And again, it's really, really good to see so many of you asking questions and on Frog. And I'm doing my best, I hope, to answer them as best I can for you. Again, please keep them filtering through if I'm not asking. And again, I will say it one final time, if there's any actual factual content that you are panicking about the night before, just give us a comment and I'll do my best, certainly by the end, to answer it for you. Okay, we're now going to look at tables, the second big component to the exams. Again, we're not needing much content, or certainly much scientific facts, to get the answers here. So we got some kind of test or trial. They're doing preclinical, clinical phases. They've got some people there testing on or particular way that they're testing on it. They've got some compounds, they've got the time taken. What's the shortest time taken to develop a new sleeping pill? Well, on this sleeping pill, the absolute shortest time, this is going to take me a year for my preclinical. Phase 1, the shortest time is 2 years. Phase 2, the shortest time is 1 year. And phase 3 is 2 years. 1 plus 2 plus 1 plus 2 equals 6 years. No science needed there. I've got to read some numbers. Again, what's the range for the number of volunteers needed to complete all the clinical trials? The range, my smallest to my largest. So I don't have any people to test here, but I've got 20, 100 and 1,000. So that equals 1,120. If I'm nervous in an exam, I'm using my calculator at this point. You're a fool not to use it, even if your maths is A star. Check the calculator to be sure. And I need my maximum range. Well, I've got myself 100, 500, which is 600, and 5,000. So my range will be 1,120 to 5,600. Reading some information from a table. The second table we're looking at is a little bit more complicated because they put some information in. So again, it's time for my highlighter. Gardeners are using weed killers to control the growth of plants. A gardener wants to get rid of daisy plants. They investigate using a weed killer. The gardeners recorded the number of daisy plants in a 10-meter squared area. They've made solutions of the weed killer. They've got different concentrations. In other words, one's really strong, one's got no weed killer in it at all. And then they've put some of that on a different patch of lawn, and they've recorded the number of plants in the areas after 2 weeks. So we've got the number before and the number after. The gardener decided results for a concentration of 20 of weed killer was anomalous. Doesn't look right. Something's not going right here. Why? Well, very simply, he's got 6 plants before he uses the weed killer, and he puts something that's meant to kill weeds down, and he suddenly ends up with 8. He gains 2 extra plants. Either his weed killer's gone mad at that moment, or there's some kind of anomalous result. Something's gone wrong. He really needs to repeat or ignore that one. But the actual answer is very simple. I started with 6, I ended with 8. So again, all we're going to do is look at simple points within tables. Okay, I've got one more table example for you, and it's this one. Again, all from last year's paper. In 2011, more than 600,000 people collected votes for the UK National Bird Survey. And for those of you that are astute, this is actually exactly the same table that I took out a minute ago, but then had a graph around it. People recorded the number of each species of bird they saw in 1 hour on one day. For example, on Wednesday at 12 AM, or 12 PM actually, so we can actually be up, they looked and they found a certain number of birds. So let's have a very quick look at this particular table. The question being, in the UK, house sparrows are more common than blackbirds. Suggest 3 reasons why this may not be true. Well, at first looking, it would appear that house sparrows, of which there are 4.1 seen per garden, are in fact more common than the blackbirds that are 3.2. But that's not all the information they're giving us. If we look at the blackbirds, 95.2% of them were found in gardens. That's versus only 64.5% of the house sparrows. So this is a positive to the blackbird. If you actually then multiply the mean number by the percentage seen for both, you find the house sparrow number and I've pre-done this one using my calculator, came out at 264.45. Whilst my Blackbird number came out at 304.64. In other words, I've got myself a bigger number for my Blackbirds. There's a second reason they might be a bit more common. The third, I'm not going to use my table for the third, but the third comes down to common sense. They looked for 1 hour on one day. Well, what happens if the cat was in the garden for that hour? What happens if it was raining in some gardens and not the others? In other words, this is pretty woolly at best. This is not that useful for 1 hour a day. It gives us a rough indication, but you could have argued any other circumstance for why it might appear to actually not be correct information. That's not a huge amount of data to look at. Okay, that's tables done. I did have a very quick question regarding the graphs that I'm very sorry I didn't quite answer. So the question was, will those kind of graphs be on tomorrow's paper? Well, obviously it's so difficult to say, but these particular graphs won't be. That being said, graphs really similar to these will be. The reason I can tell you that, well, one's a pie chart. My second one is a bar chart, a column chart, as is my third, and my fourth is a XY scatter or a line. In other words, I've got all three disciplines of graphs that we kind of do at GCSE level. It's almost certain that you're going to get graphs very, very similar to that. Okay, we're down to two final parts now. The 6 marker. I'm not really bothered about the answer here. The answer you should know, because hopefully your revision is going well. But remember, they've given us our information. We're going to be assessed on our good English, organisation, specialist terms where appropriate. In other words, this is our SPG. The question I'm not that bothered about, but it's plants responding to environmental factors, describing the growth of roots, the growth of shoots. I need to refer to a plant hormone. Do not refer to the artificial use of plant hormones. In other words, I've already got my clues here. I know the things that I need to be talking about. And the rough rule of thumb for a 6 marker is you need probably 3 to 4 good scientific points along with good spelling, punctuation, grammar. But just think about this number here. This is 6 marks out of 60. This is 10% of your paper. Do not leave it blank. So, really roughly speaking, if I was doing this and I was talking about plants, well, I'm dropping in the word auxin, and auxin gives us an unequal distribution of growth. My plant is going to respond to tropisms, could be photo, geo, hydro. And then I might be saying, well, if it's phototropism, shoots will go up. If it's geo, shoots will go up, roots will go down. If it's hydro, roots will go towards. So I've got myself 3 or 4 good points from my factual revision. I'm then ensuring I don't refer to the things that I want. I do talk about the things that they do. And I put it in a nicely uniform way. Don't write for the sake of writing. Don't write and rabbit on about this and that. Be concise, be to the point, but do check capital letters, full stops, commas, and that it flows genuinely quite well. Okay, we're on to the final part now. And these are tick boxes. These are questions that mainly come up at the start of papers. They might be used more or less in the foundation papers and the higher, but it's useful to know what they're going for. I've just had a very quick question about the multiplication on a graph. Don't worry, I'll come back to that one. And there's a couple of questions on the nervous system. So I will go back to those two in a minute. But for the vast majority of us, I'll just finish off on the tick box question. Now, I said tick boxes, etc., because these are things that unbelievably people just either forget to do or do it incorrectly. So which temperature will be most suitable for growing bacteria in industry? Again, it's a fact. I hope you're looking at 40, 25 school, and 100 is going to kill them. But lots of people will write 40. They haven't asked you to write it. They have told you to ring it. Simple, but do. When we've got tables, we're looking at receptors where there's a change in the environment. So my receptor because of sound, well, this is going to be my ear. My flashing blue light, well, that's going to be my eyes. And cooler air temperature, that's going to be on my skin. Make sure you write in the box. It's worth 3 marks. You should have 3 answers in the box. Figure 1 shows a light receptor. Pretty nasty question, this one, actually, because we wouldn't specifically learn a light receptor, what it looks like. But we're asked to use the correct answer from the box to label A. If you do that, you're correct, but you're getting no marks. They haven't asked you this time to ring it. You've got to write it. I mean, this is simple stuff, but under the pressure of an exam, things you might miss out on. So be careful. Final couple of points here. If we've got ourselves more tick boxes, again, I've got myself a ring around. So if we're looking at being affected by genes that we're inheriting, we might be unlucky and have a higher cholesterol level. We're then filling in to complete each sentence. So when we are ill, white blood cells produce antibodies. And many strains of bacteria, including MRSA, develop resistance to drugs called antibiotics. Now, you might want to get there and tick them off as you get there. You might want to brain dump, go pathogen, bacteria, virus, vaccine, dead or weakened. Hold on, antibiotics kill my bacteria. MRSA is a bacteria. In fact, they even tell me it's a bacteria there. So, brain dump, so be careful. Last couple of bits with diagrams. I wanted to show you this particular one. We're having to list apparatus A and apparatus B. Well, apparatus A, you kind of either know that or you don't. That is our inoculating loop for when we're taking bacterial swabs. But B gives us a lovely clue. It says it's a sterile solid culture medium. B, well, what do you put your medium in? It's for Petri dish. But actually, you would have got a mark for saying it's the agar jelly. You wouldn't have got a mark for simply saying it's the culture medium, but you would have actually got a mark for calling it agar in a Petri. So clues in the diagram. Here's the last question. Again, likely to come up in your paper, some kind of adaptations. I've got an Adelie penguin and a chinstrap penguin. Adelies and penguins live in the Antarctic. It's below 0. There they are. Adelie, they spend most of their time on the ice. Chinstraps like to live in the sea and around the ice. And then since 1965, the Adelies have decreased. Figure 8 then gives us some clues as to the last 50 years. Why have the Adelies decreased? Well, we've got a load of ice here and we've got bugger all here. In other words, we've got ourselves a massive loss of ice. And hopefully at that point, you're linking that, well, if they like to live on ice and there's no ice, they're losing their habitat. Perhaps they're finding it more difficult to find their food source or their shelter or wherever it happens to be that they use the ice for. So they've given us the clue in the diagram. And the very, very final question. We're looking at a method for the future production of a human. I've got a female, person A, I've got this cell P, I've got cell W that forms an embryo, I've got person B, a body cell. Use the correct answer from a box to complete the sentence. The something of cell P is removed and discarded, or there it is being discarded, but hopefully shares to you. That is the nucleus. So, ladies and gentlemen, as a main part, that's everything covered. I hope you found that useful. Do not panic. I'll go back to my original slide. Make sure you're having a good breakfast, you're eating well, you've got some lunch. Have a good night's sleep. Get ready for your exam and turn up promptly. Check your pencil case now. And when you get there, 60 marks, 1 hour, be aware of your time. Give yourself time to check. Look at the number of marks. Answer every question. Find some space. Take a brain dump where you can. And finally, with all of that, the very best of luck. I'm going to stay online now just to answer a few people's questions that I haven't answered as we've gone through. And then hopefully, I wish you adieu and good luck. So the very first question that I've had that I didn't really answer particularly was Bradley asking about the nervous system. The nervous system is one of those interesting ones. Essentially, you've got yourself a person. I like to draw them out, and perhaps that person sees someone. In my example here, perhaps there's a pretty girl. And again, what's going on here? So we're going to have some kind of stimulus. It's going to go into this chap's eye, the receptor. It's then going to go up a sensory neuron. Now this will be a conscious response. So it will go up to the coordinator, the brain. And then it will travel back down a motor neuron, which will go to an effector, in other words, something like a muscle, and we'll get some kind of response. Let me think. Perhaps this chap will wave his hand to that particular person, or maybe he might blush a little bit. So hopefully, Bradley, as we discussed earlier today, that might be a good way to remember it. You might have a slightly different take on it. I've got another question just regarding the multiplication on the graph. Really, really difficult one, this one. So if I just get to it, which question was it? It was this one. It's a nasty question. The long and short is this. We've got ourselves an 80% increase. So we're looking at what an 80% increase on 35.5% is. To put that another way, if we had a 100% increase from this, then twice what 33.5 is would be 67%. They essentially want me to find 80% of 33.5 and add it together. So 80% of 33.5 would be 33.5 divided by 100 times 80, or simply 33.5 times 0.8. That number you then add to your original percentage of 33.5 to give you a total. And I can't remember off my head, I think it was about, well, let's just do it on my calculator. Okay, so it's 60.3%. Again, if you're not quite following that, don't massively worry. It's just a couple of math skills at looking at a percentage. I've got a very quick question here as well that is asking about glucose likely to be in the paper. Do you mean questions concerning graphs and tables evaluating sugar, or do you mean content about glucose that will ask to talk about insulin and the effects of glucose? My gut feeling says they've been talking in the news, and again, you do not need to know these specifics, but I think the added sugar that we are allowed for an average human man is about 30 grams of additional sugar. And I think I'm right in saying a can of Coke has about 7 grams in it. And so they might be asking about the issues regarding this, about balanced, healthy diet, about getting all the correct nutrients. Is this too much of our simple sugars, of our broken down carbs for a healthy, balanced diet? And with that in mind, they might say, if we've got massively high blood sugar levels, what are the implications? Well, hopefully you would link that insulin is used via homeostasis to control this blood sugar, where we take it from the cells, where we take it from the blood and put it into the cells as storage. And again, if we take far too much sugar into our everyday diet, we have got this potential of causing ourselves to have type 2 diabetes. Again, this is all conjecture. We haven't seen the paper, but these are things where, because it's been quite current in the news, it might come up. Again, other current things in the news at the moment are actually in the last couple of weeks. They were talking about the problems with antibiotics and antibacterial resistance. So perhaps MRSA is another one that is, because it's current, might be coming up. Okay, ladies and gentlemen, I think I've answered all the questions that have come up. Very much hope you've enjoyed it. Wish you all the very best of luck tomorrow. And remember, get some sleep at this point. All the best.